Okay, good morning, good afternoon, and good evening. So my name is Budiman Milnasni. So I'm a professor at the University of Sydney. I'm also a member of the Science and Technical Committee of the 4 per 1000 initiative. So welcome to the 4 per 1000 science webinar. So this webinar series try to provide some information about the latest what's happening in the science of the soil carbon. So this session will discuss about soil health. So we all heard about soil health, we all heard the buzzword soil health, but what it is this soil health, and how do we really actually measuring it? So we have two speakers who's going to give insights based on their experience about soil health measurements. So let's start. The first speaker is Dr. Iêda Mendes. Iêda is a researcher at the Embrapa Cerrados. Iêda starts as an agronomy from the University of Brasilia and later pursue a PhD in soil science from Oregon State University. So she joins the Brazilian Agricultural Research Institute Embrapa in 1989. And now she is an active researcher in the Embrapa Cerrados, Brasilia. And recently she is very interested in the measurement of soil health to tell us about something about the soil. So let's start with Iêda.
All right. Thank you, Budiman. It's a great pleasure to be here with you, sharing this webinar with Beate and Cristine. I'm going to share my screen. So let's see. Okay. Well, so thanks a lot for the invitation, Budiman. Good morning, good afternoon, good evening. It's a great pleasure to be here. About 4 years ago, I participated in one of these webinar series by the 4 per 1000 initiative. And back on that time, I presented the launch of the soil bioanalysis technology. And today I'm really happy to be here again in this webinar because I'll be able to show you how the adoption of the technology, of the soil bioanalysis technology, has evolved in these past 4 years. So the title of my presentation today is Large-Scale Assessments of Soil Health in Brazil using Soil Enzymes. And as Budiman said, I'm from Embrapa. Embrapa is the Brazilian Agricultural Research Corporation. It has 46 units spread all over Brazil, and I'm from Embrapa Cerrados, which is located in Brasília, the capital of Brazil. We work in the Cerrado regions, which is the most important agricultural region in Brazil, with about 207 million hectares. And before the 70s, Brazil was not a food secure country. In the past 50 years, Brazil was able to develop a science-based advanced tropical agriculture. And the evolution of Brazilian agriculture is closely linked with the adoption of practices that improve soil health and crop yield, such as the adoption of no-tillage in about 32 million hectares, integrated crop livestock systems in about 17.5 million hectares. And more recently, we really have an increasing adoption of cover crops, mostly deep-rooted African grass, such as brachiaria, which are excellent chemical, physical, and biological conditioners. So it's another revolution in the tropical agriculture. And in line with these global concerns about soil health, Brazil adopted the soil bioanalysis technology, in short, soil bio, in July 2020. It consists on the inclusion of two soil enzymes, beta-glucosidase from the carbon cycle and erisulfatase from the sulfur cycle, as part of our routine commercial soil analysis. So soil bio is like a soil's blood test. It allows us to anticipate asymptomatic soil health problems before they reflect in terms of losses of green yield in our farms. Soil enzymes are more sensitive than soil organic matter in order to detect changes that occur in the soil as a function of the adoption of different management practices. So they work as ecosensors, radars, and they can be messengers of good news and bad news. And why we should care about soil health in our tropical soils? Well, we know that soil works like a superorganism, and the superorganisms can get sick. Today in Brazil, we have a huge amount of data showing that healthy soils produce an average of 686 kilograms more soybeans per hectare than diseased and healthy soils. So healthy soils are more productive. And also, healthy soils are also more resilient to these extreme weather conditions. So here, for instance, we have two plots from a long-term field experiment in Mato Grosso. Treatment 1, soybean was cultivated under fallow. Treatment 3, soybean was followed by brachiaria. And it's interesting to point out that in terms of chemical fertility, these two treatments were basically really similar, a little bit of more soil organic matter in treatment 3, but the point is that these two treatments, in terms of soil biology, they were completely distinct, with more enzyme activity in the treatment where soybean was followed by brachiaria. So we had a condition where we had chemically similar and excellent field plots, but biologically distinct. On the 7th year of the conduction of this field experiment, we have a really extreme dry spell during the reproductive stage of soybeans. And what happened? The treatment where soybean was followed by brachiaria produced 1.8 tons more per hectare as compared to the treatment where we had only soybean monocrop. So this really showed us that healthy soils were more resilient. And in this case, the yield correlated with soil biology and not with soil chemistry. And the reason why that soil on the brachiaria was able to tolerate, to have a better performance under that dry spell is because brachiaria improves soil physical structure. So by improving soil physical structure, the soil was able to store more water, and as a consequence, the soybean grown on those plots was more productive as compared to the soybean grown on that treatment where just had soybean monocrop with a very poor soil structure. So this shows us that soil biology walks hand in hand with soil physics, right? So the importance, it shows how soil biology and soil physics reflect in our crop yields. So really powerful connections. The choice of beta-glucosidase and aryl-sulfatase was the result of 20 years of research into Brazilian oxysols. In the list of the advantages of using these two soil enzymes are associated with their sensitivity. They are related to nutrient cycling. They are correlated with other microbial indicators of soil health, such as microbial diversity, microbial biomass, and soil respiration. They are precise, consistent, and the analytical procedures are simple and relatively inexpensive. And also, they can be performed directly in their dry soil samples, which really makes their adoption at the commercial laboratories easier. Very important, these two soil enzymes also present very good correlations with crop yield, which is very important from the economical aspect, and also a good correlation with soil organic carbon, which is the most important indicator of soil health, soil quality. And also by using soil enzymes, in addition to evaluate soil health, we can also access soil's memory, because soil enzymes are not only associated with the living component, but when the living cells die, soil enzymes can be absorbed on the clay particles and also in the soil organic matter. And in this way, they reflect the past generation of living organisms that were present in the soil. So by using soil enzymes, we can detect the fingerprint that every management system keeps in the soils. So today also in Brazil, we have a huge amount of data also that shows that soils with higher yield and glue activities are soils that can store more water, have low phytonematodes populations, have a better nutrient use efficiency, better potential for bioremediation, better food quality. So that's why they are really good soil health markers. The protocols for the soil bioanalysis is totally standardized, which enhance data quality and interpretability. And also is very important for soil health assessments. The determination of soil enzymes by the soil bio protocol follows the Tabata procedure. So in this case, we measure potential soil enzyme activities. And the good news is that within 2 months or so, FAO is going to publish the SOPs for soil enzymes. In terms of soil sampling, the procedures are similar for those adopted for soil chemistry, but the biggest difference is that for the soil bio, soil sampling must be performed at the 0 to 10 centimeters depth. So the diagnostic soil layer for soil bio is 0 to 10 centimeters. In terms of when to evaluate, is at the post-harvest, and it's very important to perform soil sample at the same time every year. In the soil lab, in the commercial soil lab, the soil handling procedures involve working with air-dry soil samples and also sieving the soil samples at 2 millimeter sieves. So following the same procedures that the commercial labs usually adopted for soil chemical analysis. A very important contribution was how to interpret the individual values of these two soil enzymes. How do I know if that enzyme number that I have for my soil is low, is moderate, or is high, is adequate? And in order to do that, in order to make this calibration, we use the same principles applied to the soil nutrient calibration. So in long-term field experiments, where we have plots with and without phosphate fertilizer, we were able to modulate soil organic matter and crop yields. And in this way, we were able to build these response curves of soil enzymes as a function of crop yield. So in order to establish the adequate level for soil enzymes, it was very simple. So the enzyme level necessary to reach 8% of the maximum crop yield was considered adequate. And the enzyme level necessary to reach, for instance, 40% of the maximum grain yield was considered low. The same way that people from soil fertility build response curves for soil phosphorus, for calcium, nitrogen, and etc. And today, we have interpretative algorithms that estimate enzyme reference values as a function of clay content. So calibration is the key word, calibration as a function of grain yield and organic carbon. Soil bioanalysis goes beyond the determination of the two enzymes. It also involves the calculation of soil quality indexes. And the conceptual model of the soil biotechnology integrates chemical and biological indicators in three soil functions. So the two soil enzymes are associated with the nutrient cycling function. Organic matter and cation exchange capacity are involved in nutrient storage. And the acidity components, base supply, phosphorus supply are associated with the nutrient supply functions. All these functions are integrated in soil quality index, and in order to calculate the soil quality index, we use the same procedures used for this method, described by Andrews, Douglas Carlin, and Stott. So basically, each indicator is scored using those standardized scoring functions, more is better, less is better, and optimal. And the unique thing that we did in the soil bio was that all the function scores and all the soil quality index, again, were calibrated in those long-term field plots as a function of crop yield and soil organic matter, so calibration. In order to make the soil bio report easy to understand for our farmers, we use a 5-color scheme following the traffic light. So the higher the value is green, and the lower the value close to the red. So that's how it works. And today in Brazil, finally, we are moving from that traditional soil analysis report based solely on the mineralistic concept. And now we have a more holistic, comprehensive soil report, which show us how is the ability of our soil to function, to provide nutrients, to store nutrients, and to cycle nutrients. So it's a new view of the soil that goes beyond that solely question of nutrient excess or deficiency, okay? So how it works? Embrapa has an innovation network, a partnership with commercial laboratories. So today we have 33 commercial laboratories in Brazil performing soil enzyme analysis. And all these commercial laboratories are connected with Embrapa through a web platform called Soil Quality Interpretation Module. This web platform makes the interpretation of the results of the enzyme activity determinations performed by the commercial soil labs, and also calculates the soil quality index and the scores for those three soil functions. And based on the combination of the scores of the nutrient cycling and the nutrient storage functions, we can separate 4 types of soil health conditions. We can define 4 types of soil health conditions. Healthy soils punctuate high on both functions. Soils getting sick have high punctuation for nutrient storage, but low punctuation for nutrient cycling. Unhealthy soils have low punctuations for both functions, and soils undergoing regeneration have low punctuation for the nutrient storage and high punctuation for the nutrient cycling. So these are 4 patterns found in nature. I'm going to show you how it works with some examples, some real-life examples from real-life farms performed by commercial soil labs. So here we have an example of soil bio type 1 report, where we have high punctuation for nutrient cycling, high punctuation for nutrient storage, so healthy soil. This soil bio report is from a farm a 1,500-hectare farm in Mato Grosso do Sul, totally under integrated crop livestock, so the soil is chemically and biologically excellent. Here we have an example of the soil bio type 2 report. In this case, we have low punctuations for nutrient cycling, but high punctuation for nutrient storage, which show us that the soil is degrading from the biological point of view. This soil bio report is from a farm, a huge farm, 22,000 hectares farm in Mato Grosso, totally under these intensive soybean cotton production systems. Here we have an example of the soil bio report type 3, unhealthy soils, diseased soils. In this kind of soil bio report, we have low punctuation for both functions, nutrient cycling, nutrient storage. So it's important to call your attention for the punctuation of the nutrient supply function in these bio reports. You can see that the punctuation for the nutrient supply is totally in green, indicating that the soil was chemically perfect, but the biological component, the cell health component, was totally compromised. So in situations, in this type 3 bio report here is from 5,000 hectares farm in Mato Grosso, and again, under intensive soybean cotton production systems. And it's important to tell you that we have a diseased soil, but in a condition where the soil is chemically excellent. So in situations like this, soil chemically corrected, many times before the soil bioanalysis, what the farmer would do? He would add more fertilizers, more pesticides, which are the right medicine, but for the wrong disease. You won't fix a soil health problem by using fertilizers or pesticides. You need to change your management practice, right? And finally, we have the soil bio report type 4, soils undergoing regeneration. In this case, the soil was degraded, it has lost soil organic carbon, so low punctuation for the nutrient storage, but the farmer has adopted practices that are regenerating the soil, so soil enzymes are more sensitive, so they detect these changes in the soil, and as a consequence, the nutrient cycling function punctuates in green. So that's how it works. Here, I show you the soil bio report for these 4 farmers, but also I show you the soybean yield average in the past 4 years in these different conditions. And as you can see, the healthy and recovering soils are more productive, and the difference between these 2 soils, healthy and recovering, in relation to getting sick and unhealthy, is about 0.7 tons per hectare. Show you again that healthy soils are more productive, but these are now under farm conditions. And this is an example of how soil bio works in the field. Here we have a huge farm in Mato Grosso, 40,000 hectares. And at your left, we have the map of the nutrient supply function. You can see that in terms of chemical conditions, the farm is excellent. But when we designed the soil health map, based on the punctuations of the nutrient cycle and the nutrient storage functions, we can see that the situation is completely different. 41% of the farm have possessed soils in unhealthy conditions, 15% of the farm contain soils getting sick. So it's a new view that goes beyond those excess or lack of nutrients, right? Soil bio is a very democratic technology. It also works for small farms. And now I'm going to show you how are Brazilian soils doing. What have we learned about soil health in Brazil after these 4 years of the adoption of the soil biotechnology? So this is the first version of the soil health map of Brazil. This paper is under review right now. And in order to build this map, we considered every municipality in Brazil as a mapping unit. Brazil has around 5,000 municipalities, and in this 1.0 version of the Brazilian Soil Health Map, we have data for 956 municipalities. And we could see that 77% of these municipalities had soils in a healthy and recovery conditions, 19% of soils getting sick, 1.4% unhealthy, and 2.6% what we call an intermediary state, where exactly we don't know if the soil is healthy or unhealthy. But this is what the kind of data that now we are extracting from the soil bio database. When we submitted the paper, our data bank had around 26,000 soil samples, And now, in June 2024, the soil bio database has more than 35,000 soil samples, and it's growing. And in the next few slides, I'm going to show you some really neat information that we are learning from these data. Here, I'm going to show you how the soil health condition is associated with soybean yield at the state level. And in order to do this, we worked with two databases, the soil bio database, where we have the soil condition by each municipality, and also with the agrivision database from Embrapa, where we have the soybean yield in each municipality of Brazil during the period 2017 and 2022. So this is the situation of Paraná State. Paraná State was one of the top 5 Brazilian soybean producers, and Brazil is the world's largest soybean producer. Paraná is a state that's about 2/3 the size of Italy. And look, based on the soil bio database in the agrivision database, we could see that as long as we increase the percentage of unhealthy and sick and getting sick soils, there is a reduction in soybean yield. And with an R2 of the regression of 0.6, so it's really significant. And this kind of behavior we also observed for other 4 great Brazilian soybean producing states, Minas Gerais, which is about the size of France, Mato Grosso, which is 1.5 times the size of Ukraine, and Mato Grosso do Sul. And out of the top 5 Brazilian soybean producing states, only Goiás did not present a good correlation in terms of soybean yield and the percentage of soils sick and getting sick. So the thing is that here in Brazil, we were able to develop this technology in small plots, in long-term field experiments. And now this technology works on the farm level and also at municipality, state, and national level. So it's really scalable, right? It's simple, it's practical. And the secret is calibration. Soil bio works inside and outside the farm gate. And now our next step is to use this data for public policies on soil health in Brazil. What the farmer does, as long as he has his soil bio reports, is very simple. If the soil is healthy, keep going. You are in the right way. If your soil is getting sick, attention. You need to change. You won't let your soil, you can't let your soil go to an unhealthy condition. If your soil is unhealthy, is sick, act with urgency, use cover crops, brachiaria, and if your soil is recovering, keep improving. So that's how we use the soil bio reports to support management decisions. Well, in order to keep tropical soils healthy and alive, we need to mimic Mother Nature, right? It's the best template, intelligent design. So we have plant diversity, minimal soil disturbance, living plants covering the soil, straw, animals, trees. That's the best way to keep soils alive and healthy under our tropical conditions. So in this way, we will move towards an agriculture based in process and not in products. All the papers that form the basis of the soil bio approach have been published in the best journals in the field. In last March, the SSA Society, the Soil Society of America, has published the volume 3 of the Soil Health series. And the volume 3 is only dedicated to soil health and sustainable agriculture in Brazil. And in this volume, and in this new book, we have a whole chapter about soil bio, how we developed this technology, how it works. And, the point is that to access soil's memory was a complex problem, but as researchers, we should find simple solutions. And soil bio is really, really a simple solution. 2 soil enzymes, soil organic matter, 0 to 10 centimeters depth soil samples, and then you can tell to the farmer if the soil is healthy, unhealthy, getting sick, or undergoing regeneration. And I love to finish this talk with this quote from Leonardo da Vinci. Simplicity is the ultimate sophistication. So the cost of a complete soil bio today in Brazil, a complete soil bio involves the determination of the 2 soil enzymes, the chemical parameters, and the soil texture. It costs $36. Well, thank you so much for your attention. This is the Soil Bio team. It's a group of wonderful researchers, students, field workers, field technicians. So this is the team behind this technology. A lot of hard work within all these years. Thank you so much for your attention. I'm going to be very happy to answer your questions and comments. after Christine's talk. Thank you so much.
Thank you, Iêda. Thank you for the wonderful talk and a very enthusiastic presentation. So we'll just keep we'll just move on to our next speaker. Our next speaker is Dr. Cristine Morgan. Cristine is the Chief Scientific Officer at the Soil Health Institute in the United States. Cristine earned her master's and PhD in soil science from the University of Wisconsin-Madison. And then she was a professor of soil science at Texas A&M in College Station. And now she is just now in 2019, she joined the Soil Health Institute, and her work is responsible for establishing research priorities to advance soil health. And over to you, Cristine.
Thank you. Thanks, Booty. Hello, everybody. Happy to be here. Good day. And what I thought I would talk a little bit about is some work that is all primarily published, but just really focusing down on like how to measure soil health and how the Soil Health Institute has decided to measure soil health continentally for North America. So essentially, Soil Health Institute, if you're not aware, is a small nonprofit research institute. There are about 30 employees, and our mission is to safeguard and enhance the vitality and productivity of soil through scientific research and advancement. So that's what we do. One of our big projects has been about, a continental assessment of soil health. And I won't talk a lot about like how we're assessing soil health right now, but I thought what I would do is kind of review some of the knowledge gained from our North American project to evaluate soil health measurements. The goal of this particular project was to figure out, what are the most effective indicators of measuring soil health from the continental United States or the continental North America and the United States. And so what we did is we looked at 124 long-term experimental sites. And what you're seeing here in this image is the location of the long-term experimental sites, and then a index showing, essentially blue is moist and cooler, and red is drier and warmer. So you can get an idea of the climatic range of these long-term sites. So we had 124 long-term sites. Each site had a paired treatment with either tillage, cover cropping, or crop rotation. And we went into these sites in the spring prior to any manipulation for biomass production, and took a measurement of the soil from 0 to 15 centimeters. And from that measurement, or from the soil samples, we looked at over 30 current and exploratory soil health measurements. I'm not going to go through all of them. I thought I would just kind of review them based on, kind of how we looked at it and help you understand how we came to the conclusion that we did. And so the main thing is we had all of these measurements, and we needed to think about a way of how we would come down to this minimum suite of effective indicators for North America. So we had tons of measurements. A lot of these measurements were decided based on blue ribbon panels that we had with experts in North America, soil scientists that had been working in the area of measuring soil health. And so we started out with these lot of measurements, and then we started thinking, okay, of all these measurements, what actually reflect soil health? And measurements that were thrown out in this decision process were like inherent properties, soil texture, fertility-focused measurements, because we're actually interested in soil functioning and not necessarily the soil fertility per se. And if we're thinking of a continental assessment, soil fertility is something that is very local with local calibrations. And we didn't really want to create a fertility calibration. So we took out measurements of fertility, and then those measurements that are under development still yet, which were a lot of the qPCR microbiome type measurements. The next thing we did is we looked at the measurements and how they responded to these long-term practices. So we had a lot of paired treatments. And when I say long-term practices, we had control and adoption of different soil health management practices. And the time that those practices had been in place was probably around 20 years, with the low end of 8 years and a high end of about 50 to 60 years. of application. So we had all these paired measurements. And we looked at, and that's the data I'm going to show you today, is how they, some of these measurements responded to these deltas, or, you know, the difference in the treatments versus control. But then when we're thinking about a continental assessment, and it was covered in the previous talk, is about price and availability. We had to choose measurements that were available in commercial labs, or that had the potential to be adopted by commercial labs and not necessarily just by research labs. And then lastly, we really looked at our measurements that had made it through the rest of these filters, about whether or not they were redundant. You know, did they provide sufficient additional detail, or are they just kind of like other measurements, so that we could really hone down to an affordable approach to monitoring and evaluating soil health. And so I just skipped to the conclusion. And the conclusion was is that we felt like the data showed us, along with the interviews and the applicability in the laboratory, that the measurements that we selected were soil organic carbon concentration by dry combustion, carbon mineralization potential, which was the 24-hour respiration measurement, aggregate stability via slaking analysis, and I'll talk a lot more about our selection of the methodology for measuring aggregate stability. And then for temperate soils, North American soils, we have a pedotransfer function that predicts available water holding capacity. If you're interested in any of these in more detail, you can scan this QR code or go to this paper in Soil Security, which references the 6 other peer-reviewed publications that we published on each one of these individual selections. But I just thought what I would do is just kind of go through, you know, we think about soil health, we think about soil functioning, and the functioning we really looked at was carbon cycling, nutrient cycling, and water cycling. And so I thought I would go through some of these measurements and just kind of share the way we're thinking about, we're monitoring soil health. We can't always actually measure the function that we're interested in measuring, but we can select an indicator of that function. So one of the first functions that I have mentioned is carbon storage and carbon cycling. And when we think about carbon storage, some of the measurements that we selected in our experiment were soil organic carbon, permanganate oxidizable carbon, water extractable carbon. And when we think about carbon cycling, we're thinking about both the, the substrate of the carbons there, but also, what is the activity of the microbes, how's carbon being cycled? And some of those indicators that we measured were carbon mineralization, either with the 24-hour incubation or a 96-hour incubation, and then beta glucosidase, which is an enzyme associated with carbon cycling. And so a lot of the data I will show today looks like graphs like this. So I will slowly walk through this particular graph. What we have on the y-axis for this graph are the different management practices that we had treatments for in these long-term experiments. So we have rotation diversity, like how over the time period that these treatments were in place, how many different crops were in rotation, including cover crops. Crop count. With diversity, we're also looking at, is it a monocot versus a diacot? Crop count, total number of different crops that were cropped in those treatments. Residue retention. A lot of these were like biomass studies where the residue was removed or not removed. Adding organic nutrients. Many times it was compost or manure, not biochar. So primarily organic nutrients associated with manure. Cover cropping, the absence or presence of cover cropping in between the cash crop. And then decreasing tillage. So decreasing tillage included if the delta of the control to the treatment was a less intensive tillage, that's considered a decreased tillage event. And so what we have is the relative on the x-axis, for soil organic carbon, what we're looking at then is the difference between the treatment and the control. The control was generally the preponderance management practice for that cropping system. And then the treatment would be adding more diversity, adding residue retention, looking at addition of manures, adding cover crops, or decreasing the tillage. And so if the data point falls on 0, that means that there was no difference between the treatments. And then, of course, if it's on the positive side of the 0, then we have an increase in that measurement based on the addition of that treatment. So essentially what we're looking at was soil organic carbon for all those 124 sites and 600 plus treatments. We can see that rotation, diversity, and crop count did not affect the soil carbon, organic carbon concentration in 0 to 15 centimeters, but residue retention, organic adding manures, cover cropping, and decreasing tillage all significantly contributed to increases in soil organic carbon across these treatments on average. Same with oxidizable, permanganate oxidizable carbon. We had the exact same trend. With water extractable carbon, a very, very similar trend. With the enzyme, again, same trend. And then for 24-hour and 96-hour carbon mineralization, a very similar trend, with 24-hour incubation being more significant more frequently than 96 hours. It was a little bit more sensitive than the 96-hour incubation. So what do we conclude from that? If we are interested in a soil health indicator for changes in soil carbon storage, that soil organic carbon is not only a direct measure of carbon, but it was just as sensitive as any of the other measures that we looked at. Water extractable organic carbon and POC had the exact same trend as soil organic carbon. So when we're thinking about what do we measure and adding measurements, do they have additional information? The answer was measure soil organic carbon, and the others do not have additional information. We looked at carbon cycling. Again, the 24-hour carbon mineralization was most responsive compared to the enzymes and to the 96-hour. Then when we looked at nitrogen pools, we looked at total N, which comes from dry combustion as well. Water extractable organic nitrogen, autoclave citrate extractable protein, ACE. And then when we thought about nitrogen cycling, again, we're looking at some enzymes and potentially mineralizable nitrogen, which is the corollary of the 24-hour mineralization of carbon, but under different laboratory conditions. And the big conclusion we found with all of these nitrogen indicators is the carbon told the story for nitrogen. Again, this is not about fertility. This is about nitrogen cycling and nitrogen pools in the soil. So total carbon and soil organic carbon are very linear related. We've known that for a very long time. If we looked at the water extractable carbon and nitrogen, also very linearly related. And same if we looked at the enzymes, the nitrogen enzymes versus the carbon enzymes, and the mineralization potential of carbon and nitrogen, there just wasn't any additional information by looking at some of these nitrogen indicators. And then thirdly, we looked at water cycling and storage. So we had some direct measurements of saturated hydraulic conductivity. We used this little Saturos. And then water storage, when we think about that, we want to know does plant available water change? And we have a direct indicator of measuring plant available water. And then we have indirect indicators of plant available water, which would be bulk density, aggregate stability, or soil organic carbon. And again, now the X and the Y axes are, well, I've switched things a little bit. And on the X-axis now, instead of having the management, we just have the different measurements that we used. And now each plot is a different management practice. So when we'll walk through slowly the first one for residue retention. Essentially, if we retain residue, we have an increase in soil organic carbon, we already know. Intact field capacity, that's an intact soil core, improves or increases significantly. Bulk density decreases significantly. So we're looking at effects of pore structures, and water stable aggregates aggregate stability increases significantly. A couple of just big general comments of things that we found. Of course, permanent wilting point and field capacity of a repacked soil, those are really measurements of soil texture, and so they're never really responsive to changes in management. So I'll just not talk about those. And then the other one, which was my favorite, is saturated hydraulic conductivity. Being a soil physicist, But the problem with saturated hydraulic conductivity, even though we had replication, was that the variability of the hydraulic conductivity was so great, we could not see a signal, especially a statistically significant signal. But the take-home message here is that solar organic carbon continued to be a nice proxy for for water cycling. But other than that, water stable aggregates was also a good proxy for water cycling. And so was this field capacity intact, was a nice direct measurement of changes in water storage and water cycling across the different management practices. So ultimately, kind of what we concluded with the water storage issue is that, again, we have indirect measurements that were super sensitive, solar organic carbon and aggregate stability. The direct measurements, again, was that intact field capacity. But in the end of the day, we decided that it was a really hard measurement to take. It's a hard sample to collect, and not a lot of labs want to do 1/3 bar or 33 kilopascal suction on large soil cores, intact soil cores. So we ended up with a pedotransfer function that was pretty well tested for North America and seemed to work. And then lastly, what we wanted to look at, because we're really thinking of the practicality of things, is we looked at comparing methods of aggregate stability, because there's so many ways to do it. And there are a lot of papers about this. Particularly what was unique about us is we added a different aggregate stability measurement that's fairly new, which is the SLAKES measurement, which is slaking by image recognition. We used the Cornell rainfall simulator, wet sieve procedure, which is the Yoder method or the traditional method where you take the aggregates and you wash them. And then we had a mean weight diameter estimate also from some sieving wet and dry aggregates. And essentially, we found all the aggregate stability tests worked pretty well. The Cornell method and the slaking by image analysis method were the most consistently responsive to soil health management practices. I don't show you all the data here, but here's just an example where the very, very popular method of washing aggregates through that Yoder method was not statistically significant for adding cover crops. So just some additional new information about how these aggregate stability methods line up. And again, when we're thinking about our continental assessment, we really like this aggregate stability by image analysis because you can do it with a smartphone. And we have re-released this slaking image analysis. It's free to download through Google Play or an app store. And we also want to acknowledge University of Sydney Booty in originally developing this app and delivering this app. And we just tested it and made it a little bit more robust. That's all. And then lastly, when we think about using these measurements across landscapes, I am not talking a lot about this, but just to give the big picture, we don't like doing indices. We like actually just benchmarking the soils in different areas and then keeping the measurements separate, because in some ways we have indicators that are changing and indicators that are not changing when farmers change their practices. So we really advocate looking at those measurements independently. And here is just like the benchmarking concept where we're going out and looking at soils under perennial systems and then soils under row crop systems, but with different management practices. And what we're doing is creating reports to farmers where for their soils, we baseline, provide measurements for soil health management practices, and also give them a reference so that they can understand how healthy their soil is today and what are some reasonable, attainable goals that they could create. And this is just an example of some of the data that we have. This is from Ontario, Canada. But essentially, we looked at about 200 farms, and we're finding, that organic carbon concentration in this particular case, of course, is related to clay content. So we can't just do, basic, looking at means. We have to detrend the means based on soil inherent properties. And so this is an example of that. And then ultimately, what we're doing is reporting to the farmers based on the detrended data. That particular farmer, in this example, the farmer's data is this red line. And they can now see how their soil organic carbon concentration stacks up to different row crop management practices, and then also up to what is the potential of that soil to store organic carbon. So just quick summary remarks. We looked at these selected indicators based on soil function, and most indicators were sensitive to tillage, cover crop, residue retention, and organic amendments. Soil organic carbon and nitrogen indicators behaved very similarly, and testing indicators in addition to soil organic carbon and total nitrogens added confidence in our results. There was no simple indicator for water movement. We really had to think about like what was doable and what's practical, because ultimately there will not be soil scientists out there taking gorgeous bulk density measurements. There are going to be people out, collecting soils for fertility analysis and other things that we want to use these samples for. And that's it. I just want to thank all of the people that were involved in this project and hand it back over to Booty.
Okay, thank you, Cristine. Thanks for a wonderful presentation. So we might just continue with the discussion. So for the discussion, we have Dr. Beata Madari. Beata is also from Embrapa, and Beata is a member of the scientific committee of the FORB 1000. Over to you, Beata.
Thank you, Budiman. First of all, I would like to thank for the two excellent presentations. It was very interesting, and both of you were very enthusiastic about soil health measurement. Well, the concept of soil health is not actually new, right? But at least in the scientific environment. However, it is a fairly recent development that it receives increased attention from governments, policymakers, from farmers and other stakeholders, given the role of soil and soil functions that support ecosystem services, including environmental conservation and improving higher or more stable yields, and eventually farm profitability. Well, not mentioning that soil per se is one of the two most important natural resources that are essential to provide food security. It is quite clear that there is the need to develop comprehensive and widely applicable soil health measurement to be able to evaluate the effect of land use and agricultural management on soil health and validate agricultural practices that improve soil health. So, well, both of you in your presentations, in your studies, you used initially a fairly diverse data set, so physical, chemical indicators, also indicators of biological processes and microbial communities, and managed to finally get to a minimum or reduced set of indicators to monitor soil health, soil quality, under the different land use and agricultural management practices. And with this, it is, well, you have soil health measurement methods that are relatively easily and effectively can be used to evaluate sustainability in agriculture and to support farmers and policymakers. But first of all, I would like to hear your opinion about a more general subject. Today there is a lot of emphasis made on monitoring, reporting and verification for soil to detect soil carbon stock changes, and specifically to be able to use this for carbon markets and gain income so that farmers could gain income over soil carbon increases. However, do you think that methods like methods for soil health analysis could eventually be more useful to indicate the sustainability of farm management, even in programs where farmers could be financially compensated for improving the quality of and the health of the soils? And Yeah, and especially regarding the role of agriculture in climate change. So through soil health measurements, would it be eventually possible to assess the potential impact of agricultural systems for climate change? I mean it the two ways, emission of greenhouse gases and also the ability of systems to remove carbon from the atmosphere. So finally, the carbon balance of agricultural systems. I know it's quite general, but I don't know if you would like to address this a little bit.
Can I start?
Of course.
I think so, Beata. We have data showing that with, for instance, soil enzymes, we can evaluate soil carbon trends in the soil. So, for instance, we can see if the soil is in a trend to gain carbon or to lose carbon, and also if the soil is in a condition of positive stability. For instance, a healthy soil where you have high enzyme and high soil organic matter, and also if you are in a equilibrium, but a not very good equilibrium with low soil enzymes and low soil organic carbon. So in this way, you can use enzymes to as like eco sensors to detect if you're in a trend to gain or to lose carbon. But it's difficult to correlate with soil carbon stock, because soil carbon stock, it will change very slowly. So you might have a soil with a huge carbon stock, but the farmer has changed the soil management, and then the soil is going to start to lose carbon. But only with in the medium on long term, you will see changes in the soil carbon stock. But the soil health indicator will be able to give you signs of that trend to lose carbon earlier as compared to the whole soil stock. And soil stock measurements for farms are very difficult because, for instance, for IPCC, you need to go to 1 meter. So it's kind of hard for them to make this measurement. So the soil health indicator, because of its sensibility, is able to detect these changes earlier, change for good and for better. And for instance, for soil enzymes, because with the most part of the work we did with soil enzymes, we have good correlations of soil enzymes with nitrous oxide emissions. For instance, soils with high iosulfatase activity have low nitrous oxide emissions. So the point is, in the soil, everything is connected, right? So as long as soil biology goes well, we will be in a virtual cycle, so that we are going to have better soil physics, better carbon sequestration. So basically, that's what happens.
Yes.
So,
Yes, Cristine. Thank you.
yeah,
Iêda.
those are good questions. I think the one thing that we often tell people when asking about carbon is, I just want to make sure, we're the Soil Health Institute, not the Soil Carbon Institute. And there's a very important reason for that, because we believe in the vitality and productivity of soils, and soil carbon concentration is not the only indicator of the improvement in soil functioning, especially in row crop management systems. And so a couple of things you asked about like MMRV for crediting farmers for their management choices or improving soil functioning. And that's the whole reason why we wanted to land on a very small suite of indicators that address all the different kind of major functioning roles of soil in biomass production. And then and we really focus not on yield, because I think it's pretty much shown that yields stay the same or maybe increase with improved soil functioning. But the important thing is profitability. And the other thing is you really wanting to be optimizing biomass production and maintaining and improving soil functioning. So for us, the optimization is keep yield the same and increase the soil functioning. And the whole reason why we created our benchmarks process is for MMRV. For instance, particularly in cotton production in the United States, buyers are very interested in tracking soil change with bales and providing premiums or some other mechanism, even if it's like marketing preference for improved soil health with whatever the agricultural product is. And then our benchmarking is kind of an MMRV, because any farmer, if they live in an area, they know, they have a map, they know what soil they're sampling. They can send off that soil health, their soil sample for soil health analysis. And then that data can go on top of that benchmarking data. And then they can show where their soil health is relative to similar soils in that area and that same climate under different managements. So we're very much, our eye is on measurement and monitoring. We also, I didn't show, but we also are doing soil carbon stock in the United States. Most of the payments for carbon stock are to 30 centimeters, not that much deeper. And what we're finding is, carbon concentration is increasing and the stocks are changing, at least in the spots where we're measuring, but we're not doing a whole carbon stock assessment of fields. We're just trying to, I think the most helpful thing with our benchmarking and carbon markets is for a farmer to be able to see what is the potential of their soil and what is that delta for row crop management. And so we think it's maybe a really good decision support tool for folks if they're thinking about entering the carbon market.
Okay, thank you very much. Well, now if I have time, Buddy, I would like to address just some very more specific questions, which is my interest, and I hope that all of those who are watching will find useful. But actually, Cristine, you did not talk about this, but in some of your publications, you mention some advanced comprehensive techniques like visible near-infrared spectroscopy for the measurement of soil health. This technique can characterize or quantify various soil properties simultaneously. So what would be the future role of these methods? Could they effectively substitute in practice the actual classical measurements, lab analysis? So, or how close are we to these solutions?
That's a great question. I think Viz NIR is the reason why we work with it is because it's an in-field measurement, and it doesn't need, a soil sample does not have to be taken back to the lab. If we talk about soil samples being taken back to the lab, I'm a firm believer. I think MIR is a very good substitute for lab measurements. And the whole reason why we are looking at or promoting the slaking by image analysis is that is already a very accessible proximal method for measuring soil aggregate stability. So I think, a thing I did not talk about is those 3 measurements were selected because I see a day where those 3 measurements are made, not in the laboratory. Someone could make the aggregate stability measurement today with their smartphone, have colleagues that are very close to respiration measurements that do not require expensive machinery. And then, soil organic carbon could definitely be monitored with MIR. It's not going to have the same accuracy, and we might need to think about our sampling methodology changing. If we're going to use MIR, we might get more samples so that we have an average. But yeah, I think that is definitely the future, and that's our eye, because in the United States, and in Canada, where we're doing this work, we're extremely spoiled with a lot of access to laboratories. But we're definitely looking towards, how can what we're doing in North America be applicable to areas where there's just not the same lab access?
Yeah, actually, one of the great barriers of soil health analysis is the lack of laboratory facilities in many parts of the world. Yes. So these techniques, yeah, they would be very useful. But Iêda, as I know, please correct me if I'm wrong, that the method that you presented, it was tested for clay oxysoils of the Cerrado, right? And for certain crops like soybean, corn, so the major cash crops, actually. So are you working on extending the scope of the methodology? I saw the presentation that, for example, you are working with integrated crop livestock forestry systems, which is a very important trend in sustainable intensification in Brazil. But for example, extending to other soil types or even other climate regions, and linked to this, and the possibility to adapt this methodology to other countries with similar climate and soil conditions in Brazil. Are you working with this, or are these in the plans?
Oh, yes. Thanks for the question, Beat. When we first released the soil biotechnology, it was calibrated only for oxysoils in the Cerrado. And then 1 year later, we expand the adoption of the technology for the soils in the subtropical regions of Brazil. And today, the technology is calibrated for Cerrados annual crops and also annual crops in the southern part of Brazil, where we have the subtropical climate conditions. But the neat thing is that even 4 years after the release of soil bioanalysis, many people that have other kind of crops, such as sugarcane, pastures, eucalyptus, all the other crops, they have also submitted their data to our interpretation platform. And it's interesting because even though these crops were not usually initially included in our studies, with the algorithms that so far we have developed, we are able to see different soil health conditions, even in these crops where the technology was not calibrated for them. So this is really interesting because many people that have sugarcane plantation, pastures, are also submitting their data. And even though the algorithms were not developed for those conditions, they are working on that. So we believe that, also very interesting, in the northeast part of Brazil, where we have a semi-arid region, the algorithms developed for the Cerrado conditions, which is like a savanna-like vegetation, also work for the northeast part of the country, regardless of the soil type, because since we are only working with the 0 to 10 centimeter soil depth, according to the pedology, my friends from the pedology, it doesn't matter a lot if it's a argisol or an axisol, what matters on that 0 to 10 centimeter soil depth is the clay content. So the clay content would be the driver for these different responses. So, and you guys were talking about MRV, measuring, reporting, verification. Now, with the soil bio database, we can track all the soil samples. So what we are really seeing for the future is payment for environmental services using, for instance, soil enzymes as metrics. Because now, based on soil enzymes and soil organic carbon, we can see these different types of soil health conditions. We can separate big farms as a function of the management system. So it's totally traceable. We can track those soil samples and we can follow their behavior all over the year. And we were talking about the possibility of these measurements being adopted in soil labs all over the world. For instance, in order to perform soil enzyme measurements, you only need in your soil lab an incubator and also a spectrophotometer in the visible light spectrum. So, which usually are equipments that are present in every soil labs. So when the soil bio was released, one very nice thing was that it could be used in every commercial soil labs in Brazil. Even in big labs, small labs, because it doesn't require it's not necessary to buy expensive equipment and fancy equipment. So I don't know if I answer your question, but after all these 4 years, the technology really has surprised us a lot. At the beginning, we were very concerned about, oh no, only annual crops, row crops, and all that. And now we can see that it's working in pastures, eucalyptus, and all those different situations that we didn't work at the beginning.
Yeah, and following up what you said, that it is actually the methodologies and certified laboratories, commercial laboratories, so it's available actually for farmers. and eventually policymakers actually use it, this method. Okay, just one last question. Well, actually, it's just it stuck with me, Iêda, for you as well. You mentioned soil memory, and yeah, that soils can maintain a better response to stress, like maintaining the memory of good agricultural practices, the result of this. But how long do you have measurements or data? Like how long this memory lasts?
You know, that's so cool. You know, we have some data showing that now using soil enzymes, we can establish a biochemical dialogue with our soils. And this access to soil memories using soil enzymes is really, really amazing. For instance, we have we have a long-term field experiment in Mato Grosso where we had only soybean under fallow. So the soil was really in an unhealthy condition. And then we decided to change, to regenerate that treatment using brachiaria as a vaccine. You know, brachiaria is that African grass with deep roots. And after the introduction of brachiaria, soil enzymes start to increase, let's say 3 months. We introduced the brachiaria on March, and in October, we already saw the soil enzymes increasing their activity as a response of the vaccine. So the soil memory, you know, everything passes in the farm. The soybean passes, the pasture passes, but the soil it stays there. So he holds the memory of everything that happened in the past. So in this experiment now, we are seeing how the soil is recovering, and we know I look like crazy, but I really think that we have now this possibility of to talk with our soils. So again, talking about the soil memory, as long as we switched from soybean fallow to soybean followed by brachiaria, the brachiaria presence in the system has already changing the soil's memory. And now, after 3 years since the introduction of the brachiaria, we have noticed this increase in soil enzyme activity, showing that it doesn't take too long for the soil memory change as a function of the different management practices that you adopted in your farm. So, you know, and of course, we are talking about the 0 to 10 centimeters depth, which is the soil depth most impacted by plants, which are the agents that modulate the whole soil functioning, right? Yeah. But it's really interesting. And I think the day that the farmers really feel that they have this capacity to communicate with their soils, for instance, using soil enzymes, powerful changes will happen, you know, in our farms, because it's really, really amazing.
Branding, yes. Well, thank you very much for your participation, for the answers, and I think, Budi, now we have to switch to the questions of the audience. Thanks.
Thanks, Beata. I think we have answered most of the questions. There are a few specific questions because I think we run out of time. So it is already 1 hour and 10 minutes. I think we don't want to pursue it longer, but I think Cristine or Iêda, they will be happy to take your questions via email, I assume, Iêda, okay? If you have any questions or to Iêda or Cristine, please send them an email and they'll be happy to answer it. So I think we need to close up this webinar. But before we go, I have a question for both of you. A simple question. So what we talk about soil health. So what do you consider as a healthy soil, Iêda?
Healthy soil is a biologically active soil, able to store water, to perform nutrient cycling, to have a better nutrient use efficiency. You know, it's a soil resilient to water stress, with a good water infiltration capacity. It's only good stuff, you know, better potential for bioremediation. And now we even have data showing that the quality of the soybean that is produced in healthy soils is better than the quality of the soybean produced in unhealthy soils. So it's all connected. So soil health, healthy soils, healthy plants, healthy planet. So everything connected.
And you, Cristine?
I think a healthy soil is a soil that's functioning well for the creation of biomass or cycling water, cleaning water. It's a very subjective question, and it's very like soil centric and human centric. So a soil is healthy if it's a functioning in a way that benefits society or nature or any of our existential global challenges.
Okay, thank you. So a healthy soil for healthy people. So thank you, everyone. Thanks for the presentation, and I'll see you next time.
Bye-bye. Thank you.
Bye.